This video is concerned with DNA and RNA. The genetic information for our body is stored in the DNA. Originally they thought that proteins were the genetic material because proteins are much more complex molecules than DNA. But now we know that DNA is our hereditary material, and it is the information specifically to make proteins. So the information to make one protein would be considered to be a gene. The genome is our entire set of genes, and that was completely discovered in the Human Genome Project was finished in 2003. And the genetic code is the exact method of translating those nucleotides into the information to make the amino acid sequence in a protein. We'll first look at the structure of DNA. DNA is deoxyribonucleic acid. We call the structure of DNA a double helix. This was discovered by James Watson and Francis Crick. DNA consists of two strands of nucleotides. Remember, the monomer for DNA is a nucleotide. The nucleotide consists of a sugar called deoxyribose, a phosphate group, and a nitrogenous base. The nitrogenous bases for DNA are adenine, guanine, cytosine, or thymine, and that's represented by A, G, C, or T. The backbone for each strand is a sugar-phosphate chain. The bases are complementary to each other. That is, adenine is complementary to thymine, therefore adenine always bonds to thymine. Cytosine is complementary to guanine, therefore cytosine always bonds with guanine. DNA itself does not consist of a molecule that's by itself. It is associated with proteins called histones, and there's quite a bit of wrapping. Just as a thread is wrapped around a spool, the DNA is wrapped around these histone proteins for organization as the chromatin condenses to form chromosomes during mitosis. Here we can see DNA on the right. On the left is a single strand of DNA. On the right is double-stranded DNA. You can see the sugar-phosphate backbone, and in the center will be the bases. Connecting the bases would be hydrogen bonds. Remember that adenine is complementary to thymine, and cytosine is complementary to guanine. Here you can see the molecule of DNA and its double helix nature. And further on down the molecule, you can see that it is associated with histone proteins, wrapped around those proteins, coiling, and supercoiling in order to form the more condensed form, the chromosome. DNA must replicate itself exactly during the S phase of mitosis. If the replication is not exact, then a mutation has occurred. What happens is the hydrogen bonds between the base pairs break, and the DNA strands unwind. The base pairs in the center of the molecule are exposed, and complementary base pairs come in and bond to those exposed bases. This is controlled by the enzyme DNA polymerase. DNA replication occurs during the S phase of the cell cycle. So here we have interphase, which consists of G1, S, and G2. DNA is replicated during the S phase. In this diagram down below, you can see that the DNA is unwinding, and these exposed bases will then bind to their complementary bases. So here we had adenine was exposed. In comes thymine. Each new molecule of DNA consists of one old strand and one new strand. This is called semi-conservative replication. The genetic code is the specific sequence of amino acids. Well, actually, it's the information that codes for the specific sequence of amino acids in a protein. And that information is in the DNA. It's actually in the nucleotide sequence. 3 nucleotides represent one amino acid. DNA has to stay in the nucleus. DNA is the reference for the protein information. And just like a reference book at the library has to stay in the library, the DNA has to stay in the nucleus. Proteins, however, are made in the cytoplasm on the ribosomes. So the DNA's code is copied and carried to the cytoplasm. This copy is an RNA molecule. RNA differs from DNA in a variety of ways. First of all, it's a single strand of nucleotides. Second of all, the nucleotides, there's one nucleotide difference. Instead of thymine, there is uracil. Also, the sugar is different. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. An RNA molecule is much shorter than DNA. It's just copied a portion of the DNA. And there are different types of RNA. The messenger RNA, or mRNA, carries the code from the DNA to the ribosome. The tRNA, or transfer RNA, brings the amino acids to the ribosome. And the rRNA, or ribosomal RNA, is a component of the ribosomes. Using the RNA molecules, we can make proteins. It involves two steps, each requiring RNA and enzymes. The first step would be transcription, that is, taking the information that's in the DNA and copying it into an RNA molecule. The second step is translation. Translation occurs at the ribosomes in the cytoplasm, and that is the formation of proteins from the RNA. So here is the process. Transcription, again, is forming the messenger RNA, or any type of RNA, from the DNA. The DNA has to unwind. Remember, this is taking place in the nucleus. It opens up, and the messenger RNA nucleotides will come in, and complementary pairing will occur. Uracil replaces thymine in RNA. So instead of T, uracil will come over and bind to the adenine of the DNA. Once that messenger RNA molecule is formed, it will move away and the DNA will come back together again. This is controlled by RNA polymerase. The short piece of messenger RNA can now leave the nucleus through a nuclear pore and go to the ribosomes in the cytoplasm. Protein synthesis requires all three types of RNAs. In our cells, there are 20 different amino acids. They are always in our cells, as long as we're eating complete proteins. Also available in our cells will be a transfer RNA molecule that is attached to each specific amino acid. The transfer RNA has a region on it called the anticodon. The anticodon is going to be attracted to the messenger RNA codon. As the ribosome moves down the messenger RNA strand, the transfer RNAs will be bringing the amino acids from the cell over to the ribosome. They are attracted to the messenger RNA, and peptide bonds will form, joining the amino acids. This is how we determine what amino acid is being coded for. So we have our messenger RNA. The messenger RNA is a sequence of nucleotides. If the sequence of nucleotides is UAC, for example, UAC codes for a specific amino acid. So let's find UAC. Here is the first letter U. The second letter comes over across the top, A. And then the third letter, C, UAC. When the three nucleotide sequence UAC is found in messenger RNA, it codes for the amino acid serine. Let's try another one. AUG. First letter A, second letter U, and third letter G. Oh, it codes for two things, the starch and methionine. So if this is found at the beginning of a messenger RNA strand, this is the code for starting the amino acid sequence. This diagram represents the entire process of protein synthesis. The DNA exists in the nucleus. The DNA has to open up, and a complementary strand of nucleotides will come in and form messenger RNA. Messenger RNA is different from DNA. The messenger RNA will separate from the DNA and leave the nucleus through a nuclear pore. The messenger RNA will be associated with a ribosome. This is the start sequence here. Every other 3 nucleotides, the codon, codes for a specific amino acid. In the cell will be transfer RNAs, each one attached to a specific amino acid. On the transfer RNA will be an anticodon that is complementary to the codon of the messenger RNA. So after the start codon, the next 3 nucleotides, that codon specifies a specific amino acid. For example, if it were codon 2, GGC, it would code for glycine, and glycine would come over. A peptide bond would form between the amino acids. This is providing more detail on the different sites on the ribosome for amino acid chain elongation. This formation of the protein is called translation.